Believe it or not, your microbiome in your gut is just as diverse as this little plot of land here. There's various trees, various plants, various insects, various animals that all contribute to a unique ecosystem specific to this spot of the world. And your body is very similar in that it's unique and individualized to you. If all of us are different, what can we do to understand telltale signs that there are issues? What can we do about it, and how can we conceptualize what is needed for our body personally? So we talk with Dr. Alex Mohr, who is a scientist and researcher in gut health and the microbiome. And then not only that, but what can you do to better understand your own physiology, your own biology, and what is needed, what kind of nutrients your body requires to perform at its best. And interestingly, we also get into the metabolism and how the metabolism interfaces with the microbiome. Over 50% of the molecules called metabolites in the metabolism have in some way been modified or synthesized by the microbiome, so it's a really good look into the metabolism and how that interfaces exactly with this microbiome to create this unique picture that is you.
Today is a very germane topic to my research. Profiling the gut microbiome is one thing, but really understanding how it influences our health and improving people's quality of health is really at the forefront of what I'm trying to do and what we're trying to do at Theriome. So it's going to be a deep dive today. We're going to go over some really new science, particularly when it comes to the types of metabolites that we're able to profile coming from the gut. So I hope there's some really good takeaways, and I'm excited to get right into it. So I'm going to go ahead and share my screen. This is our research and science team. I'm right there in the middle. Obviously, I have a bit of a beard matching our CSO currently, but we are all about the science. Specifically, my role is supporting, of course, all things R&D, as well as spearheading our eventual pathway into microbiomics, or exploring the microbiome and actually forming testing around that. So this topic, of course, we're going to talk about the microbiome, the area of research that really excites me. So I'm really excited to get into this presentation. So the gastrointestinal tract, or more commonly called the gut, is central to our health. It's a canal that runs from our mouth all the way towards the other end. It's about, depending on the person, 20 to 30 feet long. And it's really responsible for digestion, absorption, and elimination of food and waste in our body. There's many organs that are interconnected, such as stomach, obviously very important to digesting foods, a lot of protein, as well as other accessory organs like the liver, the gallbladder, the pancreas. But when we look at kind of the central interactions when we profile the metabolism, or the metabolites coming out of the gastrointestinal tract, a lot of that's occurring in the intestine. So small intestine, of course, we have shown here. You can see there is a lot of absorption occurring of very important nutrients. Less so in the large intestine, but there is the most concentrated microbe, there's the most amount of microbes concentrated in this area. So it's actually very, very important. And when we are normally profiling the gut microbiome, you may have taken a gut microbiome test, for instance, you're actually sampling that particular microbiome. You're not really getting an accurate reading of what's going on further up, say in the upper small intestines, or there are even microbes existing in the very harsh environment of the stomach. Now, most people at some point in their life have gastrointestinal distress. That can express itself in many different ways. Gastric or acid reflux, for instance. You can have gas, you can have diarrhea. It's a very, very common problem. Now, it may be quite innocuous. It may not really be problematic, or it could have a major underlying condition. I have some listed here, such as Crohn's disease, colitis, celiac disease. These diseases can be debilitating for people. It's, of course, we all laugh at, poop jokes and things like that, but these are serious issues. And if you're experiencing symptomology or you're having these problems, you really want to get some type of testing done. The other part that I really want to make clear, because the gastrointestinal tract, as I stated initially, is so tied to how we're interacting with our nutrition. But 70% or approximately 70% of the body's immune system actually is surrounding the gastrointestinal tract. So we understand that it's critically important, particularly as you're developing as a human, as an infant, to train your immune system, get you ready to interact with the world. But also, a lot of things that are coming in, it's, you really have to make sure that you're giving your body everything that it needs to make sure that that tissue is not being overloaded. It's called the gut-associated lymphatic tissue, or GALT, and that it's working with you, working with your gastrointestinal tract. It's having a symbiotic relationship with the microbes, the microbiome, which we'll get into. And,
Do you mind if I interject a question or two as we're going throughout this?
Sure, yeah.
So the first question is, you mentioned that if people have these symptoms that they would want to consider testing, but I heard the acid reflux, but what were some of the symptoms that you were kind of thinking that, hey, that's an indicator that you should look into this further, that you should probe further and understand what's going on?
That's a great question. So there's pretty obvious ones. For instance, if you're experiencing blood in your stool, that's a pretty good indication that you need to go to the doctor, you need to go to your physician and get that checked out quite immediately. Other symptoms can be, I guess, a little bit more latent, a little bit, maybe you just kind of brush them off as not being, a big issue. Kind of the frog boiling in water analogy. You don't really understand how bad it is until you eventually get those issues resolved. A lot of it can be food intolerance. People are just not consuming the diet that's really in tune with their body or conducive to the microbes that are inhabiting, particularly the lower regions of places like the colon. So that would be gas is actually a very, very common problem. And I mean, it sounds, again, not to make light of these issues, it sounds kind of silly, but if you're chronically bloated, it can take away your concentration. It can result in some embarrassing social situations. Other ones, of course, are constipation. I've consulted several people that they're having one bowel movement per week, and they're just getting tremendous buildup. And the level of discomfort that they're going through, again, while we make light of this, we make light of these types of issues. It's terrible. They can't exercise. They have trouble sleeping. It can really, really impact your health. So, when you're starting to experience those issues, there's not like a specific, one test that's available oftentimes in traditional medicine. You have to go get this large profile done. Stool tests can be very revealing, very important. Oftentimes, though, if you're going through traditional routes, it's going to take a long time to get results. Other issues can include SIBO, where you're getting overgrowth. You're going to have to go get some breath testing for that. They're actually looking at the gases that are coming out of gastrointestinal tract. So it can be very complex to actually identify and hone in on what the underlying condition is with a lot of these. Others, like I said before, are a little bit easier, but because there are so many, this is a really important area. And as I said, so many people suffer from these issues that we really, I think, need to put a lot more emphasis in truly understanding our gut health and all the factors that really play a large role in shaping a healthy gut and a healthy gut microbiome.
Yeah, it has so much impact. And you highlighted that 70% of the body's immune system is located within or around the gut. And then I know this is a little bit off track, but I know you've done some research on fasting. Is that in part why fasting has such a positive effect on the body, is because it alleviates the need for our immune system to be acting and working in that area?
Absolutely. In fact, as simple as it sounds, we actually in our publication that we put out a couple of years ago, talking about some of the newer research that's coming out in this area, we termed it gut rest. It's the ability of just having, because if you think about it, most people are walking around in a fed state for 12, 14, 16 hours a day. Their gastrointestinal tract is constantly working. Um, of course, all the other associated organs like the liver, the pancreas, they're also working. But for many people, breaking that habitual intake cycle can be very beneficial. And you're absolutely right to bring up the immune system. There's also certain microbes that may be getting fed that we don't want there. Fasting might be a mechanism to maybe reduce those populations and encourage the growth of more healthy bacteria, as well as actually encourage the growth of mucus that surrounds the inner layer of our basically our gut barrier. So if you look at the gut, you're taking a cross section of the gut. So we have these cells along the border called the brush border, and they're very, very important, but it's just one layer of cells, and they're interacting with all these microbes that are, kind of in the center of that lumen. So it's very beneficial to have a nice, healthy mucosa, this mucus that surrounds the gut lumen. And if we're constantly not eating the right things or eating too much, it can actually decrease the mucus layer. And that can really be problematic for these microbes to now encroach upon that delicate brush border and really cause some inflammatory issues. So fasting, we're still exploring this area, but it seems to be encouraging mucus growth, basically.
And we'll probably do a separate video just on fasting. And I know Dr. Jasby and possibly you did some of the research as well, but on vinegar, I believe as well, right? The use of vinegar prior to a meal and various things. Fascinating subject. We'll get into more in another video.
Absolutely. We as humans are hosts to billions of microbes, mostly in the lower portions of our gastrointestinal tract. The actual biomass, this has been debated, but it's approximately around 200 grams. So it's something that you can actually kind of fit in your hands. If we were to take out all the microbes from your gastrointestinal tract, that's a lot. I mean, that's pretty significant. As you alluded to at the beginning, Micah, of this talk, it's estimated that approximately half of the metabolites that we find in our body have been synthesized or modified by these microbes in some way. So maybe 20 years ago, we had no idea just how much these microbes, the impact that they have on our own health, our own metabolism. So, a lot of these companies that are profiling the gut microbiome, that's good. That's a great place to start. But to truly understand the impact that it's having on us, the human, we really need to also look at the metabolites, and hence why we feel here at Theriome that metabolomic profiling is so important. So in terms of how unique the microbiome is, it's pretty well understood that everyone, once you're in adulthood, your microbiome, the different types of gut bugs are about as unique as your fingerprint. So everyone has their own unique microbiome. Everyone also has their own unique metabolome. We see that metabolite expression is extremely individual, particularly when we're looking at the metabolites that are produced by these microbes. Now, in terms of more traditional blood markers like cholesterol, maybe inflammatory markers like C-reactive protein, these are generally pretty tightly grouped in terms of the variability that we see. With the microbiome, and particularly the metabolites that we get from like the fecal sample, or even some of those metabolites that cross over into the blood, very, very variable, very, very person specific. So this is important because this ties into the types of function, the functionality of these gut microbiomes and how they're impacting our health. What does the microbiome actually do? Well, in terms of the gastrointestinal tract, it makes sense if they're all residing in that area, that they're helping harvest food, improving gut function, reinforcing our gut barrier. They can help protect against pathogens, but we can also have pathogens reside in this environment, which we can talk about a little bit later. They produce a tremendous amount of different types of metabolites. In fact, they help us synthesize vitamins, such as vitamin K, that we absolutely need for and is essential to our health. As well as I mentioned earlier, boosting the immune system, influencing different organ systems, particularly those that are close to the gastrointestinal tract, but also those that are far away. We're understanding now how impactful particularly metabolites related to tryptophan are interacting with our brain, this so-called gut-brain axis. Also our muscle tissue. There seems to be a lot of interaction with the microbes and how we're actually adapting to exercise. Very interesting topic that I hope to discuss in the future. And then more basically, when we come look at metabolic health, we're looking at traditional markers like your blood glucose, your lipid panel. We see that microbes, particularly related to bile acids, have a tremendous influence on how we're responding in terms of managing these macronutrients like carbohydrate and fat. So when we look at the colonization of our gastrointestinal tract by these microbes, really it starts after you're birthed. Now, there's some debate as to how that happens, and maybe there's some colonization happening beforehand, but the main event is birth, basically. And it's best actually if you're birthed vaginally, because that way you're coming through the mother's own microbiota in her vaginal canal, and a lot of those microbes basically inoculate the newborn and take up residence in their gut. And the reason that this is so important is going back to that GALT tissue, that gut-associated lymphatic tissue, because now we're introducing these foreign agents into the body, and it's actually training the immune system. So this is actually a good thing. Microbes were viewed for thousands of years or for hundreds of years as being something that we should sterilize and avoid. But as we've come into this last few decades, we really understand how important these microbes are for really training our immune system to handle future events as we come in, as we mature and come into adulthood. But once we come into adulthood, the microbiome becomes quite stable. Now, there are factors that can influence it, particularly what you're doing on a consistent or semi-consistent basis. So when we think about what the main traits of the gut microbiome, which truly is an ecosystem because there's so many different types of microorganisms, you can almost kind of analogize this to like a rainforest, for instance. Generally, the more types of species that you have, you're going to have increased diversity. This is seen as a healthy trait of the microbiome, because if there is an assault, say that you take a heavy course of antibiotics and you reduce a lot of the population of the microbes, if you have increased diversity, you have increased functional repertoire of the microbes. They can, if you kill off some, others can come take the place and help support that the functions that were lost, in comparison to something that has reduced diversity. If there is an assault on environments like this, it's very hard for them to rebound and come back. And this is why we find in a lot of different types of disease states, such as ulcerative colitis, various forms of cancer, obesity, diabetes, we find reduced diversity. So really there's this push to try to find a way to increase the beneficial types of microbes that we're seeing, but also the overall diversity that we have in that particular environment. Now, as I said, there's a lot of different factors that go into shaping the microbiome in adulthood, but really the main driver beyond antibiotics, which we can have that discussion where we kind of want to limit our exposure to those medications as much as possible when we come into adulthood, and even in childhood. But when we talk about really the main mover of shaping and modifying the microbiome, it is diet, far and away. There is individual variation, but it's estimated about 5 to 20% of the composition of our microbiome is affected or influenced by diet. And going back to how we're shaping the microbiome and what the microbiome is actually able to do, this, of course, is affecting a tremendous amount of metabolites that are coming in from the gut as we're absorbing food, as these microbes in the colon are interacting with different types of larger metabolites and breaking them down. Those come into circulation and then have all those downstream effects that I was talking about before. So really we see one of the main levers in which we can influence the gut microbiome and then influence our health.
So you as an expert in this area have done a fair amount of research and talked with a lot of people in regards to the microbiome and gut health. What are some of the things that you've seen over the years that you would say, yeah, that's definitely, in regards to diet, something that you want to stay away from?
So in my approach, I really believe in individuality. Everyone has different responses. So I never have complete no's to certain things. I have to see how someone's going to react. Now, in general, I can make some generalities. Going back to being in a constantly fed state, I'm of the opinion that that's not ideal for health. I don't think that we should be, first thing we do when we wake up is eat. The end of the day, the last few hours, eat. I really think we need to view eating events as exposures that can actually, like every time that you eat, you can almost view it as, is this going to be an exposure that is going to be helping me, or is it going to be hurting me? Because our body still has to handle that. We're going to be producing reactive oxygen species to digest everything. There could even be, maybe we're not eating the best, at the best places, or we're not washing our food. Like there's potential that we could get a microbe that could come in and cause some major issues. So I think we really need to view food as, these discrete exposures, and be regimented with it in a way that we're almost regimented with exercise. Because what we're finding out, particularly with, the identified clock genes in our own body, how literally each one of our cells is tuned to the circadian rhythm, the day, the oscillations that we get in the day. So too are our microbes. So, when we're feeding ourselves, we're also feeding our microbes, and we're also affecting the signals that they're putting out into our body for good or for worse. So I would definitely like to talk more about intermittent fasting and even things like the form of food, which I'll get into a little bit later in this talk. So, in terms of moving the needle, as I said, diet is really responsible for a large part of shaping that environment. But there's, of course, now others that are becoming quite popular. Antibiotics, I already mentioned. We know that probiotics, prebiotics, symbiotics, all these different formulations have definitely found a place in the market, as well as what we call fecal microbiome transplantations, FMTs. So the idea is that you take a stool sample, you create a slurry from a healthy individual, someone that would have a healthy microbiome profile, and then basically you would consume it to repopulate your own gut with that new microbiome, which is supposed to have a healthier benefit and outcompete what you currently have in your gut. Now, this is something that really needs to be done under the supervision of a physician, beyond the obvious issues that you're going to deal with in terms of, contracting some type of infection, microbe through infection. There's also issues of finding compatibility with the donor, how you're actually inoculating your own microbiome, and making sure that you are actually a good candidate for this. So I'm not advising this. I'm just simply bringing it up that it has been popular for quite some time. Probiotics are also a viable option. However, most people, the way that the market is right now, for good or for worse, is that you don't really know what you need. You're going to something like Costco or whatever, and you're buying, and you're just taking it, and you're hoping for the best. It's not really targeted. And I think this is really where tests like the Aristotle are going to revolutionize this space, because if we're able to profile someone's metabolism, we're able to profile someone's microbiome, we can understand which types of therapies, which types of microbes, which types of specific microbial strains are going to be best for that individual, rather than just indiscriminately taking a product. So in my mind, coming back to what I mentioned earlier, in terms of where we should be putting our attention to, while probiotics and some of these other exotic supplements might be beneficial, the prime mover is really going to be diet for most people. And we know that, again, going back to an adult microbiome, as I said, it's very, very stable. But when we rapidly change someone's diet, this is a pretty quintessential study published back in 2014 from David et al., where they looked at taking in either a heavily plant-based diet for a number of days, highlighted there in green. So that would be the plant-based diet to those graphs running down to your right, and then an animal-based diet, or the plant-based diet's to your left, the animal-based diet is to your right. Sorry about that. So we can see in just a couple of days that the composition of the microbiome drastically changed. So we are able to affect the populations in our gut very quickly, just based on diet. Same with with energy intake. If we're if we're going through a caloric restriction, or say that we're eating too much, again, we can affect the types of microbes, the population of these microbes that we're seeing by profiling.
And this is over how long of a period of time?
So this particular study was done for 10 days. So they did have a run-in period, which is, if you look to those graphs, that's that white part. And then the actual diet intervention was 4 days. It's that part in green. So we're seeing massive changes in just 4 days, not even a week's time.
And this isn't so much about plant versus animal as much as signifying that, hey, you can have a pretty rapid change in diversity in the microbiome exclusively through food.
Exactly. Yeah, exactly. And, this study is cited a lot, but I don't think people truly appreciate just how big of a change that these researchers observed here. And I mean, it's just speaking purely on anecdote. I mean, how many times have you gone out, maybe to a restaurant or something, and eaten something that you shouldn't have eaten, and the after effects that you felt the day after? I mean, we know this. We know this kind of intuitively, and this is what we're seeing based on these really well conducted human clinical trials. So this brings us again to, I think, a forgotten aspect of a lot of nutrition research is we're so hyper focused on the types of nutrients that we're bringing in, the amounts that we're bringing in. We forget about the cadence. We forget about, again, what I was talking about with those exposures. How often are we consuming something? Are we really paying attention to, time in between meals, when those meals are consumed? And I think this is a really untapped area for people to focus attention to, at least in the mainstream, um, on fasting and meal timing. So
Even
there,
myself, I eat relatively randomly. There's really no set schedule for when I'm eating and what I'm eating. I very rarely eat before noon, um, and I infrequently eat between noon and 4. But beyond that, there's really no set schedule. So this is all educational to me as well in regards to maybe there should be some consideration because, like you said, there's a circadian rhythm to our body that we might have that affects sleep. And there is some factor as well, not necessarily on circadian rhythm. I'm not sure exactly what you're alluding to in regards to that, but um, that our body recognizes the cyclical patterns that we have, and so that eating food has a pretty big impact, or eating at a certain time of day has a pretty big impact on that.
yeah. So the one thing that we're finding time and time again is the body generally likes routine. The microbes seem to like routine. There's debate over, you know, the best time to be eating, the best nutrients, like um, when those should be consumed. Of course, there's factors that are going to all affect this. Exercise is a big one, or physical activity. But there's many approaches available. There's, like I have listed here, some adhere to what's called alternate day fasting, where they're fasting completely for one day, another day they're eating, then they're going back into a fast. There's modified fasting regimes. There's time restricted feeding. In fact, a really interesting area, um, looking at the interactions with these food these foods as signals and the circadian rhythm, is actually cutting off feeding periods earlier in the day, where you're actually more in a fasted or time restricted eating state later. Even people like Brian Johnson, for instance, from um, of social media fame for trying to reverse aging, say he could slow down aging. I'm not sure about reversing, but he adheres to that type of eating schedule, and is seeing some good benefit, which we find in the literature as well. Of course, there's religious fasting. We forget about how food has been viewed for thousands of years. Whether it be observing some religious event, trying to become closer to God, or even go through periods of grievance. This has been really important to most societies throughout history. Now, they likely were not viewing it in terms of, what they're getting, the benefits that they're getting from nutritionally, but the fact that they were doing it likely had some benefit to their health. And we see this still practiced today with, for instance, Ramadan fasting, or even with other Western religions, like Catholicism with Lent. We still see things like that practiced. Now, in terms of the effects that we see in the microbiome, this is still an emerging area. But as I highlighted earlier in the talk, there actually seems to be this mechanism where, and we see here in the figure there, you see those little rectangles. Those represent the brush border cells. So these are the cells that are lining, as I said, the inner layer of the gastrointestinal tract. And on top of that, or within the lumen, as I said, sits that mucus layer that we want to encourage the growth of. When we are participating in fasting, or we're consuming a diet that's high in polyphenols, so think of Polyphenols, a good example would be the different colors that you get from eating different types of fruit and vegetables, for instance. Or also going through periods of caloric restriction, we see increase in abundance of a particular microbe called Akkermansia muciniphila. And basically this microbe is very special because it's kind of considered the steward or the keeper of the mucus layer. It resides in the mucus layer, and it's kind of like the steward, as I said, in that it's like the lawn keeper. It consumes mucus that is degraded, time for it to be recycled, and then it spurs signals from the goblet cells that actually produce this mucus to push out more healthy undegraded mucus. So we get this nice turnover effect. And then this keeps us nice and healthy. It keeps us nice and regular. And it also limits the exposure that we're getting from some potentially pathogenic microbes interacting with the brush border to then signal the immune system that I talked about before, that the galt tissue. So we can actually, through these different mechanisms, these second and third level effects, is we can get reductions in overall inflammation in our body.
And
now,
that was a 2-day fast that you had there, correct? So is that 48 hours, or is that I discontinue eating Monday night and I don't eat again until Thursday morning?
So we are actually in the process of publishing work that looked at 2 full days. So technically it wasn't 48 hours, it would be 64 hours. So so say you were coming into a fast, you're going to fast Monday and Tuesday. Sunday, you would stop eating at 8 p.m. and you wouldn't resume eating again until you came to Wednesday at 8 a.m. So I believe that's 64 hours. I'm not too quick on the mental math here at the moment. But yeah, we saw, and we'll be presenting that research soon once that's published, but we saw some very remarkable effects in the microbiome, and actually the metabolism, which we were able to profile as well. Now, in terms of the top 10, everyone likes top 10 lists, so I'm listing these out here. The main gut challenges based on the literature is, not surprisingly, as I've been talking about, dietary factors. Now, a lot of this relates to dietary habit, so diets high in processed food, refined sugars, unhealthy fats is actually a really large one, and we can get into that a little bit more, as well as very low fiber diets, which can be problematic for some people. But in some instances, we actually find that reducing fiber for periods of time can be beneficial. It depends on the situation, and depends on the circumstance. And this comes back to the importance of profiling that person to really give a more accurate recommendation or set of modalities. Stress and anxiety, actually we find this to be really high on the list. Psychological factors like depression can actually affect your gut health, and this is occurring through the gut-brain axis. This can also lead to symptoms like abdominal pain, altered bowel habits, and even exacerbate issues like irritable bowel syndrome, IBS. So it can be a chronic condition. Medications, antibiotics is an obvious culprit, as are nonsteroidal anti-inflammatory drugs, protein pump inhibitors, some antidepressants actually, and that makes sense if you're thinking about the signaling that can be occurring with the gut-brain axis. And a lot of these medications, it's not that they're directly affecting the gut tissue themselves. It's oftentimes they're affecting the gut microbiome, throwing off the balance of the gut microbiome, something that and I hesitate to use this word too much, but it's dysbiosis. The reason I hesitate to say dysbiosis is that implies that we have a healthy state figured out, and a healthy state is really kind of dependent or very person specific. So there's not like one healthy microbiome that we can hang our hat on. It depends on the person, it depends on the situation.
So, what would be a more accurate terminology that you would use to describe somebody who has a poor microbiome or poor balance of the microbiota?
So usually, my recommendation would be to say disease associated or like a health associated, but you're usually tying that to some type of factor or condition. So, for instance, if the person has Crohn's disease, that's a Crohn's disease associated microbiome. One area that I'm very interested in studying right now is an athletic microbiome. So we do see some similar traits, but it depends on the type of athletic endeavor. Um, so it's usually I usually give it a conditional label.
So, is the frame of thinking transfer? So, like dysbiosis is a term that says, like, hey, your gut is out of balance. Is kind of the current thinking in academia going, sounds like much stronger towards an individualized approach where it's saying, hey, we don't understand like the ideal microbiome because there isn't one. So, therefore, it's individualized for everybody.
Correct. And this is where I think ultimately this type of personalized testing is going to be so important because the National Institute of Health has been talking about precision medicine for a long time now, and we haven't really realized it. We understand, I think, or at least they understand what steps need to be taken. Obviously, a deeper phenotyping of the person or a better global assessment of that person's biology. And I would argue that the microbiome has a seat at that table. And then the appropriate cadence of when we actually perform that sampling. So in that way, we can really tie in, different forms of, different organ metabolism, circulating what we're having in circulation, what we're having in our gut, and tying it to different issues as we're going over time. And that's, I think, really how we're going to identify, okay, for this person, we find that this diet is really working for you. It's moving all your markers in a good direction. We're seeing, these particular microbes, they're producing good metabolites for you. We want to encourage their growth. You need to eat X food, or you need to fast for X amount of hours, etc. Uh, in my mind, this is really where this is really the future of healthcare, and that's why I'm excited to be involved with a company like Theriome. Now, going back to some other issues, I kind of alluded to gut microbiota imbalance. So we use that term dysbiosis. Now, the issue with having a gut microbiota that is not in line with your own physiology is it can lead to gut symptomology, inflammation. So there's actually on the outside wall of many types of bacteria, there's these little signaling agents called lipopolysaccharide, or more commonly endotoxin. And because there's a high turnover of these microbes in our gut, I mean, think about a life cycle of a microbe. We're talking about, in some instances, a couple hours, a couple days. There's a high turnover. So they're constantly lysing and breaking down. And if I have a lot of potentially pathogenic or microbes that carry a lot of this LPS, the issue is this endotoxin can cross over if we're not having a very strong gut barrier. We don't have a good mucus layer. We don't have really the integrity of those cells that I was talking about. Those little endotoxins can interact with that tissue. They can cross over. They can come into circulation, and through some downstream events, interact with the liver and cause meta-inflammation. And we actually find this state very much tied to diseases of inflammation, like diabetes, like certain forms of cancer, like obesity. So this, while the terminology I'm not a huge fan of, like with dysbiosis, having an imbalanced microbiome is not good. Basically, we it goes beyond just having gut symptomology or bad digestion. I mean, this is literally having a global effect on your body potentially.
And is this primarily isolated in the diversity of bacteria, or does it go beyond that?
It goes a bit beyond that. So it has to do with a type of microbe that the reason it's called gram negative is when we do staining of microbes, basically we can see, if it has a certain characteristic. So it's kind of arbitrary, but generally these microbes are more associated with pathology, meaning that they're they're not really doing us much favor residing in our gut. Now there are exceptions, obviously. We generally find, for instance, that someone consuming a Western diet where they're consuming a lot of refined processed foods, high in sugar, high in fat, and fat's not necessarily bad. It's usually the type of fat that becomes problematic. We see elevations in these type of microbes, and we actually can correlate the level, the abundances of these microbes and the amount of LPS that we actually find in circulation. We actually have a test for this that we do quite commonly here in our lab. And yeah, we see this correlation. So we know what the imbalance is caused by. And again, going back to something as easy to fix as diet, it's really, not to be a broken record with talking about diet nutrition. It's just really one of the prime movers where we can really affect to a large extent a lot of our issues, in my opinion. Now, in terms of diet, I mean, another main issue for a lot of people is food intolerances, allergies. Now, there's those that we can diagnose, such as lactose, gluten. Others are the FODMAP. So these are fermentable oligosaccharides, disaccharides, monosaccharides, and polyols. There's also those that are a little bit more difficult to detect. And really, this comes down to doing some more granular testing, but it can be triggered by things like food additives. Someone may have a sensitivity that has been developing for quite some time. And going back to what I was mentioning earlier on, like trying to pinpoint some of these issues, it might be difficult if all you're experiencing is bloating, abdominal pain, diarrhea, and nausea, but that doesn't mean a problem or a sensitivity isn't in existence. Now, others like infections, a lot of this, like if you have a bacterial infection or say food poisoning, or even a parasitic infection, generally, if you're going to a physician, they're going to be recommending some medications that are going to be antimicrobial in nature, which can be problematic for the gut microbiome. Other issues are autoimmune. So we know celiac disease, for instance, Crohn's disease, ulcerative colitis, autoimmune gastritis. These are issues that are going to require a bit more than just basic lifestyle interventions. They're going to require generally interaction with a physician, and potentially medication depending on the severity. Like ulcerative colitis, for instance, is a great example. There could also be structural abnormalities. Very interestingly, the appendix, people kind of thought it was this really, innocuous organ that was just kind of a remnant that we didn't really need. But now we're finding actually the appendix harbors biofilms encapsulating a signature of our own microbiome. And when people get sick, actually, or say that they had a heavy dose of antibiotics and they killed off a large part of the microbiome, a healthy appendix can actually almost be viewed as an internal probiotic dispenser, because it will actually push out those biofilms into the environment to help repopulate the gut. So people that don't have an appendix, had it removed, might be at a disadvantage if they have large reductions in their microbiome repopulating to their prior, presumably homeostatic, healthy level of those particular microbes that was part of their digestive tract.
And do we know the mechanism that caused the appendix causes the appendix to become inflamed and potentially burst?
So there's a lot of different factors that are involved with that. One of them, you know, it's related to food just getting in the wrong place at the wrong time. I mean, our bodies aren't perfect. You know, a good example of a condition related to this is diverticulosis. So generally, when you think about like one of the main actions of the gastrointestinal tract, it's peristalsis. It's this kind of massaging movement to have things pass through. Diverticulosis develops when we get pocketing, where this um, tissue is straining and we actually get pocketing and these little outcrops from the intestine, and things can just, you know, there's a lot of material moving through. The things can just find their way in. They can get inflamed. They can cause diverticulitis, which is an inflammation of those pockets. And the problem with that, of course, is that they can burst, and then, you know, it could, you know, cause a septic event. Similar things could be happening in the appendix. The anatomy could be a little bit off, where it would be more conducive to those types of things happening. I'm not a gastroenterologist, so I can't speak to every single factor. But I think, you know, one of the main issues is finding a diet that's in tune with your physiology. Like, for instance, if you're getting diverticulosis, you may not be consuming enough bulking agents in your diet to give your musculature proper tone, so it's not having to squeeze out, um, you know, these small little pencil-like stools, basically, which we know can really promote that type of event occurring. Other issues are hormonal imbalances. We know that women, particularly during menstruation, pregnancy, menopause, they're going to have issues potentially with gut motility. So how well we're passing things through. Water balance, which is also very important. Sensitivity to pain. And then also some of the more common symptoms like bloating, cramping, things of that nature. And then finally, lifestyle factors. So we can pretty definitively say that excessive alcohol consumption, not very good for your gut health. Smoking, not really good for your gut health. Some of the items that we were talking about before, Micah, such as regularity of consuming food, those exposures, that's very important. And physical activity. Physical activity seems to be just an overall elixir, as we all well know, for just about every system in our body. In terms of the signs of dysfunction, again, going back to the top 10 lists, and I'm not going to go into these in depth, but discomfort is an obvious one. Changes, if you're starting to see changes in bowel habits, particularly, we actually have a particular scale, a very simple rating scale. And I encourage everyone to actually go look this up. It's called the Bristol Stool Scale. This is used in clinic all the time to basically look at, it's basically a proxy for transit time, how long something has been going through that particular stool, has been in process through your body. You don't want things to be in there too long. And that's generally where you'll see a very dry, almost sausage-like or very lumpy stool. Or you don't want things to be in there too quickly, right? And the obvious one would be diarrhea. So you want things to be kind of right in the middle. And again, not to, you know, when we talk about the gut health and microbiome, it always kind of devolves into a poop talk, unfortunately. But that's important, and you want to actually see if you're falling into a good range. So we generally want to see between a 3 and a 4 on that scale. And unfortunately, I didn't bring a picture here, but I encourage everyone to go look at that. And then you can kind of track and see things that you're eating that are encouraging those types of scores when you have a bowel movement. Other issues, of course, are weight changes. Like I was mentioning with that one individual that I was counseling, or they were having a bowel movement once every week. They were having pretty significant weight changes based on that. And we can understand, because there's a lot of buildup. Others include chronic fatigue and weakness. These are a little bit more difficult to tie specifically to the gut, but with profiling, I think we can help get there a lot quicker in terms of what we're able to do with our testing ability. Nausea, vomiting, of course, and related to the upper gastrointestinal tract, you can have issues like acid reflux, bad breath. That can also relate, particularly if we're having some overgrowth occur, like SIBO. We can actually have gases coming up through the esophagus and coming into our mouth, and you can actually smell it. There can also be, depending on different issues like autoimmune conditions, you can have different skin issues, joint and muscle pain, as well as mood and overall mental health. Now, going back to like really what we can hang our hat on for solving a lot of these issues, diet is an obvious one. Identifying and limiting foods that might cause problems. For instance, if someone has a problem with the FODMAP type foods, maybe asparagus is, they have to remove asparagus, or maybe there's certain types of beans that they just simply can't eat. They just have a really bad reaction. Stress management, of course, we know is very, very important. Regular exercise, sleep hygiene, and a lot of sleep has to do with how regimented we are, not only with our daily activities, but also with our food. We know that going back to the circadian aspect, if we're consuming food too late in the day, certain types of food, this can have a massive effect on how we're actually going to do falling asleep, staying asleep, and various issues tied to that. Limiting antibiotic use. Antibiotics, whether they're packs that you're taking from your physician or just various household cleaning items, we just need to be very mindful and careful of that, because generally they are overused, and they can cause some pretty drastic changes in your gut microbiome. Different types of medical treatment, depending on the severity of what you're dealing with. Supplementation. I am a big fan of dietary supplements. I'm just a big fan of dietary supplements used for the right in the right instance.
Yeah, with precision.
Precision, absolutely. Hygienic practices, that kind of goes without saying, right? And I think critical to all of this is regular monitoring. So, a lot of people go out and get these microbiome tests, and I think a lot of what's out in the market has come a long way, and a lot of tests are impressive what they're able to profile. And some of the recommendations they're giving, I have some issue with how granular they're getting. We can talk about that at another time. But most people, when they go get their microbiome tested, they're just doing it one time. And that's not really, in my opinion, going to be helping you out that much, because it's a snapshot in time. We don't know if you're really looking to change the microbiome or leverage it for your health. You really need to get monitoring done on some type of regular cadence. Just like if you go to your doctor and you're getting regular blood work, we want to see where your levels are at to see if modifications are necessary. I would view sampling the microbiome in the same way. And in the future, we're actually looking at bringing a product to market to address that. Now, this segues very nicely into what's currently available to look at gut health. Now, beyond some more, I would say, archaic survey type data, the Bristol stool scale is an example of that. There's also a rating scale called the gastrointestinal symptomology rating scale. Those are good. They're a good place to start, but really to get deep into understanding gut health, we have to get a little bit more maybe intrusive, in terms of looking at the stool. So, if you are having some pretty significant issues, your doctor might recommend you get a fecal occult blood test. There are different immunological markers that we could look at in a FIT test. Stool culture. This is particularly important when we're looking at things like parasites. However, I will say that sequencing has come a long way now. When we're actually profiling the gut microbiome, we have this technique. The kind of the gold standard technique is metagenomic, meaning that we take all the DNA that we get from the sample, break it up into little pieces, put it through our sampler machine, or our sequencing machine, and we're able to identify the different microbes. We're actually also able to identify now, with increasing precision, different types of parasites and pathogens. So maybe in the future, we won't have to rely so heavily on culturing for those types of issues.
What you're seeing in the market currently, how many of the companies or tests are using state-of-the-art equipment that is up to date with what you're talking about?
So in terms of metagenomic profiling, a lot of companies have actually started to adopt this. It is more expensive. Obviously we're talking about much larger libraries in comparison to some of the older methodology, such as amplicon sequencing. However, that's not to say that those aren't still valid tests. It's just depending on how in depth you want to get. I would say the market is headed in that direction, and there's some good tests out there. I certainly can say there's some really great tests, and that's being adopted more. The issue comes in how they're interpreting a lot of that. So just identifying the particular microbes, that's good, but we need to take it a step further. And then we need to take a step further after that. So the first step to take after we profile what microbes are there is what they're doing, right? If we don't know what they're doing, it's not really helpful to us. So we need to know their function. And then after that, the next step is, okay, we have what their function should be. What are they actually producing? So this is where, in my opinion, metabolomics or profiling small metabolites really comes into play, because now we've actually confirmed to a large extent the functionality of those microbes, because ultimately it's not the microbes themselves that's important. It's what they're producing that's interacting with us that's important. So going back to some of the other tests, colonoscopy, this is something for older individuals. Everyone, once you reach that certain age, you should be doing that, at least to satisfy your physician. There are breath tests available. Interestingly enough, if you're lactose intolerant, there is a test that they'll have you take. It's very uncomfortable. It involves you drinking 50 grams of lactose. So it's uncomfortable, because if you are lactose intolerant, then you're obviously going to have issues. And one of the main ways they confirm this, beyond you just likely having trouble at the commode, is collecting gas that you're expelling. And they're actually able to look at a ratio of gases. It's a hydrogen breath test, and they're able to confirm that you actually have lactose intolerance. They can also do breath tests for something like SIBO, or small intestinal bacterial overgrowth. There's also imaging available. Of course, this can get quite expensive if you're doing something like MRIs, maybe have some kind of anatomical abnormality. Those, of course, would be warranted. Swallowing tests. This generally has to do with older individuals that might be having some maybe neurological issues, and they're not able to intervene with those muscles. There also is some emerging technology with taking a capsule and having the capsule doing an endoscopy. Also potentially looking at a capsule that could collect different microbiome samples at different sections in the gut microbiome, like the small intestines, or upper regions of the large intestines, to get a better snapshot of the gut microbiome. Those are still in process, and I'm not aware of any reliable technology out on the market currently. Obviously, food allergy and sensitivity tests have been around for quite a while. But really what's been underutilized has been metabolomics. So profiling these small metabolites that we're able to collect in various different samples. And that kind of leads me into what we're able to do here at Theriome, leveraging metabolomics. Now, metabolomics, I mean, we could look at pretty much any sample. I could look at fecal samples. I could look at blood. I could look at urine. Blood is really a excellent place in terms of assessing someone's overall global health status, because this is what's constantly in circulation in our body, and it really gives us a high sensitivity snapshot of what's going on. This is using our technology. We use gas chromatography mass spectrometry. This is a well-established technology in PubMed. I mean, the studies that have backed this up over the last couple decades, as you can see here, are immense. So this is not some secret technology. It's just vastly underutilized in medicine, primarily due to the throughput nature of metabolomics and just the archaic system, the way that the system has kind of just established itself over the years, really relying on these contract laboratories that will profile standard blood metrics, and also the culture of diagnostics and interpreting blood tests. It's a very large, slow moving ship, and getting things to turn is it's hard. But metabolomics, the really wonderful thing about it is we're able to profile so much. So it's extremely insightful, particularly when we're able to wed that with bioinformatics, which again, over the last couple of decades has just really come into its own, pulling out health signatures and increasing interpretations. So now we're not talking about a physician having to go through pages of a lab report and decipher what's going on metabolically. We can actually use our sophisticated bioinformatic pipelines to very quickly pull out health signatures and then very quickly give recommendation based on those. So we leverage, as I said, a metabolomic test, GCMS. Our current test is actually using whole blood. So we're not just looking at plasma, we're actually able to get a picture of the entire blood sample. And this gives us access to 126 metabolites, central for carbon metabolism, which represents the majority of metabolism in your body. It really gives us a great picture of your health. Now, I'm not going to list out all 126 metabolites, but here I'm just showing you a few, and you can see some that are, of course, pop out right away as, you've seen these before. Citric acid, for instance, palmitic acid, some of the amino acids such as valine, and one that we're going to talk about today, right there, number 90, L-tryptophan. So before I get into that, now looking at 126 metabolites is excellent. But if I was to give a layperson their levels of 126 metabolites, they're not going to really be able to do very much with that. The power of this test really comes into condensing or pulling out signatures from this profile for relevant health domains. And we have 12. We have aging, we have nutrition, we have inflammation, mitochondrial, cardiovascular, reproductive, liver, gut, neurocognitive, integumentary, so referring to skin, hair, nails, environmental toxicity, and mutational load. So obviously, in the purpose of today's talk, we're going to be looking a little bit at what we're uncovering with gut health as a domain. And we have a very simple rating, from a 0 or from a 1 to 10 to basically give you an indication of where your gut health sits. And what we'll be looking at today is tryptophan metabolite. So this is one component of what we're actually able to profile in gut relevant metabolites that we find in circulation. Now, tryptophan is interesting, and we're able to actually profile it with very, very high sensitivity. Here's actually one of our pieces of equipment in our lab here in Phoenix, Arizona. This is the gas chromatography mass spec that we run a lot of our samples on, is actually able to detect tryptophan. I'm not going to, in the sake of time, I'm not going to go through all the ins and outs of this particular protocol, but it's well validated, and this work has been published and is fully transparent. So tryptophan, yeah, why is tryptophan relevant to gut health? And when I go over this, people don't really understand it at first because it's an amino acid. It's one of the 20 proteogenic amino acids that we find. It's found basically in all organisms, ranging from bacteria to plants to animals. It's very ubiquitous. The interesting thing about tryptophan is it is an essential amino acid, meaning that our bodies cannot synthesize it. We must obtain it from the diet. And most people are getting in, if you're living in a Western country, generally people are getting adequate amounts of tryptophan. The general requirement is 3 milligrams per kilogram per day. So this equates to about 200 milligrams. Really the important part about tryptophan, why we need it, is it is a precursor to so many different types of neurotransmitters, hormones. I mean, I can name two very important ones that are related to brain health, neurocognition: serotonin, melatonin. We, of course, know melatonin is a hormone that is produced by the pineal gland in animals. So going back to what we were talking about with the circadian rhythm, this helps regulate sleep and wake cycles. Serotonin, a brain neurotransmitter, also important for blood clotting. It's a neurohormone actually found in many different tissues throughout the body. So tryptophan is critically important for just base level functions of our body. Now, interestingly, when we are, say we have a steak or something like that, when we're digesting that and we're breaking down those proteins into amino acids to absorb in the small intestine, most of that is getting absorbed, but there is a small amount of ingested protein that is not absorbed by the small intestine, and it actually goes to the colon. Now, the colon, as I said, really has the most dense amount of microbes. So anything that goes there, nutrition-wise, is going to interact with that environment. And that's interesting because bacteria, certain types of bacteria, actually like protein, and they can work with protein. They can catabolize it, and they can basically feed off of it and create different types of metabolites. So a class of these metabolites that I really want to highlight here is indoles. Okay, so depending on how much tryptophan is coming into that environment, different types of bacteria can interact with tryptophan. And I mean, there's many different types of indoles. There's different indoles that are attaching to things like ethanol, indole ethanol, indole propanoic acid, indole acetic acid, to name a few. And these are really important. We don't have to go into all the different classifications, but just understand that these are really important based on the interactions that they're having with the host. So again, we're going back to that brush border that I was showing beforehand. So if we look at that, if the tryptophan molecule there at the top of your screen, so this would be in the lumen of the gut, right? And all these different interactions are occurring and creating these indoles that are then interacting with the brush border cells to actually have a positive interaction. So what types of positive interactions are these? Well, they help increase mucosal homeostasis, which we talked about. Gut mucus, very important. They help decrease intestinal permeability. So each one of these brush border cells are linked together like in a chain kind of, and they have these tight junction proteins. And if we get a degradation of those tight junction proteins, we actually get what's called increased permeability, where there's the separation. So things can start to come through that we don't want to have come through, like the endotoxins, those pro-inflammatory molecules I was talking about earlier. So here we see a mechanism for actually bringing those tightly together and making sure we don't get that crossover. Another really important one is inducing the release of glucagon-like peptide 1, GLP-1. So GLP-1 we know is very popular right now in terms of appetite suppression, through drugs that are currently out on the market like semaglutide. Its indication is for diabetes, but people are using it for weight loss. But here we have a natural way to actually increase GLP-1, which we know suppresses appetite, insulin secretion, and can also slow gastric emptying to an extent.
Essentially, we were talking about the fact that there are multiple ways to look at gut health, right? You have the microbiome test, which you have developed one that is scientifically just about ready from a business end. It hasn't been rolled out yet, but we're kind of looking at later this year, maybe beginning of next year. But the idea here is that the metabolism has a direct correlation to gut health. So in this example with tryptophan, this is an essential amino acid, which is something that we take from outside of the body that we need to consume. And it creates all these interactions with the gut in the form of these indoles. So we can track essentially how our gut is performing in just this specific example by measuring these indoles through the blood.
Yes, and the important thing to understand here is the microbiome is the mediating agent. So right now with our offering, we don't offer a test, as you alluded to, or we have it ready. Basically, it's not out on the market, but we don't have something to profile that in between. But what we're seeing with the metabolites, we do have. So again, I mentioned that the important part with understanding, what's going on from the microbiome, microbiome aspect, is not the microbes themselves. I mean, obviously, we want to have a person-specific health-associated profile, but it's really what they're doing, how they're interacting with our metabolism. And here we see a perfect example. Now, there's different ways that, depending on what we're detecting with these levels, to help kind of increase gut health. And I'm just going to go over some of the really quick heavy hitters. We know that certain probiotic supplements, certain probiotic strains, such as Bifidobacterium longum, this is actually a very common strain that you can find. So there's no, that's another issue with a lot of what's out on the market in terms of probiotics, is there's all these different exotic strains coming out. But again, if you don't know what you need, it's really, who knows how helpful it's going to be. And maybe you just need a really simple remedy. Maybe it is something as simple as a very common, generally inexpensive strain like Bifidobacterium longum. Also, as I was talking about before, and we're going to have a completely separate talk for this that I'm really excited to go over, intermittent fasting. We can actually, if you think about what is coming into the colon, where a lot of this transformation is occurring with tryptophan, if you're doing intermittent fasting, you're not only having external food sources come in, you're also, different cells are sloughing off throughout the gastrointestinal tract. Those are coming down to the lower regions, interacting with the microbes. You're also giving your gut a chance to maybe repopulate some of the microbes that actually like to recycle and work on the mucus layer, which also has their glycoprotein. So they do have protein in them, obviously. So here's another avenue which we can actually help this conversion and this turnover in the gut. There's also some really directed dietary supplements available. Cardiovascular exercise actually seems to be a very robust way to increase these levels, as well as eating high quality protein, which surprisingly, with a lot of people that we deal with, while they think their diet is really good, protein, interestingly, not everyone has that box checked off. Depending on, there's a lot of health belief that comes into this. But we do know the 20 amino acids that we're getting through diet. We do know the essential amino acids that we need. but also how someone is reacting to those particular proteins. They may not do well. They may not do well with more plant-based proteins. They may do better with more meat-based options. It's really understanding how they're reacting metabolically to better attune their body to get the type of proteins that they need, and then like for instance, in the case of tryptophan, deliver these amino acids to the gut to produce things like these indoles. So those specific recommendations, I alluded to the bioinformatic pipeline that we've established here at Theriome, and really the game changer. What has made our system so powerful has been the idea of digital twins. And I'm not going to get too in depth with this. I know we're running a little bit over here in time, but digital twinning is powerful because it is personalized. Oftentimes when we get different diagnostic work done, different blood work, we're comparing ourselves to other people. When really you need to be comparing yourself to yourself in your most optimized state. And we can see that kind of visualized here with this graphic. If we kind of think about the digital you as the most healthy, the healthiest Micah can be, right? Doing all the things that is in tune with his physiology, making every single right choice throughout the day. We can visualize his health trajectory, his health score as that green bar there moving over time versus reality. We know that we don't do things perfect, either by choice sometimes or unknowingly, right? We just don't know what's best for us because we're not getting this granular type of profiling. So here we have these two trajectories, the digital self and your real self. We can see that this gap forms, obviously, and this gives us a chance to actually come in and intervene and move the needle in terms of what's actually going to work for that person. And in terms of like the actual process of digital twinning, we're taking that metabolic profile and running simulation after simulation on it to see what works best in terms of the recommendation that we're going to make. And maybe, Micah, like going back to the protein type, if we profiled you, which we have done, so I'm not going to say I'm not going to spill the beans and say all your recommendations, but maybe you do better with more meat type proteins versus plant-based proteins. We can get really granular and understand exactly what that person needs to do to really optimize their health.
A quick interjection and we'll get right back to what Dr. Mohr is talking about. But if you are interested in some of the science and what we are doing over at Theriome, we are doing multi-omics layering, which is the forefront of medicine. I do truly believe this is the next level of healthcare, bringing in personalized intervention, coupling it with large amounts of data, using AI to do some pattern recognition, and then our scientists going in and doing further tweaking from there. So if you want to learn about the state of the art of what we are doing, you can go to the link that you see on the screen right now, www.therio.me, and you'll get access to our book, some of our sample reports, and further information and just better understanding of what we are doing. It's truly cutting edge and can make a massive impact in somebody's life. So I'm in this because my brother got cancer and I wanted to better understand what we can do to help him. That was 10 years ago. So over the course of time, this has really spurred into different companies and different things that I've been doing. And I really believe that this is a way to impact healthcare on a very large scale. So I invite you to join us to dig in, to learn more about what we're doing and be a part of that. So go to this link that you see on the screen, enter in your email. You'll also get the opportunity to schedule a time with one of the biologists on our team, Dr. Jasby being the one that does the most. And you can have a one-on-one discussion with one of the biologists and better understand exactly what it is that we're doing that can drive your health to the next level, whether you're sick and you want to get better, or you're already pretty healthy, but you want to dial it up. So you want to perform better as an athlete or what have you. So I invite you to better understand precision medicine and how this can truly impact your health. So again, go to this link and back to the show.
So we talked about the measurement of tryptophan and the indoles that are basically created from this tryptophan essential amino acid. What are some of the other things that we're looking at that pertains to gut health in the blood?
Yeah, so we're actually looking at different amino acids. Histidine is really an important one. We know that has to do with the integrity of the gastrointestinal tract. A lot of the cells, like the brush border cells, really rely on this particular amino acid. So really, with our gut health test, I would say the prime thing that we're looking at is the integrity of the gut, which we know, as I was saying too, like they are really keeping a lot in. If we think about the, on parity with your own genetics, we have the same genetic material in our gut, and the only thing shielding it from interacting with our own tissue, which we don't want directly, is the brush border, is the mucosal layer, and then of course the protective immune system. So having the integrity of that system as robust as possible is obviously very important, and that was our prime consideration in providing a profile for this particular domain. Now, I'll be fully transparent. We're not capturing every single possible thing we can, simply because we're not measuring directly, or at least right now, with our current blood test, we're not measuring the fecal sample. We're not measuring the microbiome. I think the power is going to come in when we onboard that, because my vision is to pair those together, the results from those two tests together. Then we can get a really, really good idea and become very specific in terms of gut health.
Do you mind if I pull up the sample report that we had gone through and utilized in the past?
Sure.
Of the microbiome? So I feel like that could be potentially helpful. And if you have a hard stop, we just let me know. So this is, a sample report of the gut panel that you had provided of the microbiome. And so with the metabolism, right, we're looking at the gut permeability, how things such as tryptophan and the indoles might affect that, and some other various things, that are in there, but it gives you an overall gut health score, but it's not breaking that gut health score down into further granulation with each of these domains. So within here, you would see the stability, diversity, balance, probiotic panel, pathogen panel, digestion, gut barrier, and I believe we are taking off disease potential, but there's these various things that gets much more granular. And I can scroll through here if you like, if you want to do a quick overview or just talk about it for a second.
Yeah, absolutely. So in terms of how we look at the gut microbiome, it's important to understand, unlike the metabolome, where we're looking at chemical structure, we're looking at actual living things. So we treat this as an ecosystem. And we, you'll see a lot of terms that we're actually borrowing from ecology: environmental stability, diversity. These are terms used in looking at different types of environments. In my talk, in my slide deck, I showed you pictures of the rainforest, for instance, which I think is a very good analogy for the types of diversities that we kind of envision in our head when we talk about, okay, we want a very diverse microbiome. What does that look like, right? Now, stability is important and oftentimes lacking in these tests, because just like we think of our own physiology having this kind of homeostasis, right? A state of being, you achieve, say, adaptation over time, say you're exercising or whatever, you're becoming healthier, you establish a healthy, steady state. What we don't want to have happen is to move from that steady state into a disease state. So we can think of the same, analogize that to the microbiome, thinking about if we achieve a good, stable state, we don't really want it to shift too much. If we show variability, tremendous variability or instability in the microbiome, that's generally associated with a pathological issue, pathological condition. So having, like in this particular example, high diversity and high stability, those are generally considered great traits to have in a microbiome. So I would say a more health-associated microbiome. Now, moving beyond the ecology, like the basic ecology metrics, we look at, if you scroll down one page, the profile. So these are the microbes. Now, the microbes themselves, that's good, because this is kind of tells us, who's there, profiling the functionality, which we have in this report, what they're doing. That's good, but wedding it with the metabolome, then that really tells us, like, okay, this is actually how they're impacting us. But going back to those ecological metrics, if we have high stability, we have high diversity, and we see a beneficial profile of microbes known to be associated with health states, and a good functioning microbiome, meaning, okay, we're seeing a lot of short chain fatty acid producers. We're seeing a lot of microbes that are associated with anti-inflammation. We're seeing a lot of microbes that are associated or expressing a lot of biosalt hydrolase, which is important for creating secondary bile acids that are very important for signaling and improving our glucose and our blood lipids. We're seeing those type of profiles. Then we're really like, wow, okay, this is a really health associated microbiome. So it's really breaking this down into little pieces and then bringing together this story. And that's why this report, it's almost kind of formed like a narrative, because you can't have any one piece telling the entire story, as you were kind of alluding to with the gut health score overall. Like, if we have high stability, but our balance is really low, or we have high pathogens, then it's all context dependent. That may not be a good thing, right?
So how do you see the metabolomics, looking at tryptophan and the other various metabolites, interfacing with a test like this one? This one is the stool sampling, which is pretty simple. It's like a wipe, like toilet paper almost, but then you take a small piece of that, drop it into the collection piece and send it off. So it is very simple to do. With the metabolomics, we're looking at the blood. This is looking at stool. How do you see these two interfacing from a practical standpoint for somebody looking at these reports?
Well, so when we conduct these tests, we also give out a survey. So the survey is absolutely critical and fundamental to informing us, because, I mean, beyond giving basic, just like a patient intake form when you're coming to see a physician, having basic understanding of age, condition, diet, things of that nature. When we start to look at the profile that we're getting in from the metabolome, we know the person's diet, for instance, and then we look at the microbiome. It's like, wow, we understand, based on the metabolic profile, where the diet could be improved. And we see some of these signaling metabolites that we know could be very, very important for improving the gut integrity. When we profile the microbiome, then we can then look at the particular microbes that we want to increase to help encourage the expression of those metabolites. So now we can see, okay, from metabolomics, their metabolism does very, very well with this particular type of diet. Here's the specific types of foods that they need to be eating in relation to their gut health to bring up the populations of these particular microbes that feast on this particular type of digestive product from food, because you're mostly getting products of digestion when they're coming down into the colon, interacting with the mass microbiome. And then we can encourage the expression of these metabolites to reflect in the blood metabolome. So now we're getting signals, we're getting co-occurrent signals. And this is really where multiomics shines, because as we know, when we're looking at different blood tests, it's all like kind of a patchwork. Like, a doctor may look at your CRP levels, your inflammation, and he sees, okay, you're very inflamed. Okay, looking here, you're obese, you're diabetic. So he starts forming this network. We can do the same thing on a much grander scale when we have thousands of microbes profiled, we have hundreds of metabolites profiled, and we get co-occurrent signatures. And we can really start to see through things like network analyses, even leveraging AI like neural networks. Okay, these multi-omic signatures, here's what we need to do to pull up these different health metrics, and here's what's connected. That's really where the power is going to come in. And we can do that with any other system. In the future, as we onboard these other tests. Things like, and I don't want to get too far into systems biology, but if we look at your genome, for instance, you're not really going to be changing that very much. There is epigenetics, obviously, you can have some shift, but what you're transcribing, transcriptomics, proteomics, the types of proteins that you're making, we can profile all these and start considering multi-omic signatures to see exactly where and pinpoint what's going to be the most precise, intervention or precise modality to really move the needle on a health domain. When we talk about precision medicine, I think really to be precise, we need to be holistic, and that means serving as much as possible in terms of someone's phenotype.
Yeah, and I love the mission of what we're doing is in trying to make that accessible, easy to do, and then communicated in a relatively simple way to understand, right? So for somebody that just takes the test, that they can get an overall viewpoint and vantage point of what's going on in their body and what they can do about it. But there's also, I mean, you've collected so much data, right? If you're doing the microbiome, the metabolism, the transcriptome, proteome, genome, there's so much data that you can go into from there. And that is somewhat of an issue that I've noticed, is that people that are trying to get this holistic viewpoint, because that is understood, right? That the power of multi-omics shows us more. However, people will go get a genetic test, then they'll go get a blood panel for proteins, then they'll go get a panel for the microbiome, all from separate companies, but it's not translated and consolidated into a simple to understand report, because now they're kind of just looking for the redundancies between those to see what is going to potentially have the most impact. But when you're not looking at them from one spot, that might not be the correct viewpoint. And people have gotten, they get further insight, but there's still a long ways to go in that respect.
Yeah, you hit the nail on the head, Micah. It's translation. And, based on the space that we're operating in, I think a lot of people, particularly based on the condition, if they're really struggling and they're really looking for reliefment of, a certain issue, they're going to get all these different tests. And it can be really, it can be really a daunting task to do it themselves and pull these different signals. And sometimes they may have, their own personal bias, and, getting everything under one house and performing this integration and not having the siloing. It just gets me so excited talking about it, as I know you're very excited talking about it. And I look, we don't have all the answers right now in terms of, getting down to the absolute most granular level. Like, okay, you need to, clip your toenails on Monday and you need to comb your hair on Tuesday, all that kind of stuff. But, what I see in the future as we bring all this together and we build out this massive database, we're only going to be getting better. And it's just really exciting to be part of this team and being part of driving the science forward. And I think the ultimate goal here is to help people. And that's, being sequestered here in academia and doing research is nice, but, being able to take this technology and put it out into the wild and start helping people, that's really powerful.
I'm exactly in the same boat there. I'm going to switch topics a little bit, tell people how they can get a test if they're interested in understanding further about their metabolism and subsequently gut health, but also the other 11 key health domains, like you said, the aging, nutrition, mitochondrial score. It's a really powerful test to be able to speak to those things and understanding what's happening in your metabolism, but not only metabolism, how that is directly correlated to these other domains of your health. Something we didn't get into in much depth was the metabolic health association. So as you alluded to, is we can detect signatures of issues in the body relatively early on for many different things. So I believe there's 440 that are profiled in there now, correct? Or mapped out into this test? Do you know the exact number?
Yeah, we keep increasing it. I believe that's accurate.
Yeah, so that would be the various issues that can populate, and that can be advantageous to understand. Are we trending in a direction that is positive or negative? What populated in this test that might be an issue to probe further, and some of those things. So did you know, Alex, that we're doing a leap year discount
I'm sorry?
We're doing a leap year discount. I kind of just made it up, but it's a good reason to have a discount. So we're doing 20% off. I'm going to show you guys how you can get a test if you want to, and get a better understanding of your health and what you can do about it. So this is the website, which is www.therio.me. This is the full report. So this has access to everything that we talked about today. And if you use code GUT, G-U-T, it's 20% off, but only because of leap year. So it expires on March 1st. So if you want to get the test, you go to this page, get the full report. You can do a subscription, which is every 90 days, or you can change that to every 180 days. Or if you want to do a one-time purchase, add to cart, and then I'll show you guys exactly how to add in this discount. So you just go over to here, and then you type in GUT. That's going to apply a 20% discount from $650 down to $520. It's even less expensive if you do the subscription. So yeah, that's how you guys can get started with this and contribute not only to the science and the mission and what we are doing in bringing in these multi-omic layers and understanding exactly what is happening in the body and how we can pinpoint it and what you can do about it, but also getting a really good depth of information on what is currently happening in the metabolism, how that pertains to the gut, and these other various factors. So thanks so much, Dr. Mohr. It was a pleasure to speak with you and to be able to get some insight into what you understand about this. And thank you so much for sharing your wisdom.
Thanks for having me, Micah.